target period of the
It's my pleasure to introduce Professor Charles Brenner, who is the Roy J. Carver Chair and Head of Biochemistry, as well as the founding co-director of the Obesity Research and Education Initiative at the University of Iowa. Charles has his bachelor's degree from Wesleyan University and then his PhD in cancer biology from Stanford. His research interests are in the areas of biochemistry and molecular biology, with specific and continued interest in the metabolic control of gene expression. So he has forays into structural biology and bioinformatics. His research program has been funded by the National Institutes of Health, the Beckman Foundation, and the Burroughs Wellcome Trust. Let me just say a few words about the town hall format. So today, Professor Brenner will tell us about his vision for interdisciplinary research and scholarship, both in general and also as it applies to this executive director position. And then we'll open up the floor to questions. So please join me in welcoming Dr. Charles Brenner.
Thanks very much, Todd. Can I be heard? I think the microphone is on. Thanks, Todd and Susan, members of the search committee, and all of the folks from Georgia Tech that I'm meeting yesterday and today. I'm really honored to be considered for this position. In fact, I would say that to be in the conversation with outstanding scientists and engineers such as M.G. Finn and Andres Garcia is really very flattering to me. So my goals for this hour are to introduce myself, to tell you a little scientific journey that we've taken in NAD metabolism that has been clinically translated, and then to talk about the opportunities here, leaving time for your questions. Also to note that there seems to be some resemblance between our representatives here, both in color and in their personality types, so their aggressivity, I don't know what it is. So, quick disclosures, I'm going to be telling you about some technology that has been licensed by Chromadex, and I also consult for cytokinetics. So it's we've covered a lot of miles over 38 years, and I'll begin by saying that I went to Wesleyan. I got a liberal arts education with a major in biology, and I did fly genetics there, but also did a lot of debate and theater. And I mentioned debate because my college debate partner is here in Atlanta working for Turner Classic Movies. You know, in 1983, I had the quaint idea that you go to college to not be a burden to your parents. So I had a biology degree, went off to the San Francisco Bay Area to work in biotech, Chiron, DNAC, I had hair at the time, and started learning not only technology and science, but also the teamwork that exhibits in industry, where you have fully formed PhD scientists that can work together on a common problem. And in fact, we noticed in those years that biotechnology always has a bioengineering component, because you can get something to work at the laboratory bench, but you've got to be able to scale it up and control the supply chain and purify things at scale and worry about regulatory things that are well beyond what you deal with at the bench. So stayed in Northern California for graduate work with Bob Fuller in the biochemistry department at Stanford, where I purified an enzyme called KEX2 protease. Don't have time to go into it, but it was a wonderful time there. So at that point, I had yeast molecular biology and yeast genetics from my industry background. I had enzymology from my graduate work, and I decided to return to the East Coast at Brandeis, working in this lab up here with Greg Petsko to learn X-ray crystallography. And so that was a very successful postdoctoral stint. And despite the 5 years I spent in industry, I had a 4.5-year PhD and a sub 3-year postdoc, so suddenly I'm a faculty member in 1996 in Philadelphia with an F1 project, and I basically took this thing that I had in Greg Petsko's lab and turned that into a program that related to nucleotide binding proteins. In those 7 years at Jefferson, I also took a leadership role in the Cancer Center and got involved in faculty recruiting and mentorship, building programs in structural biology and bioinformatics at the Kimmel Cancer Center, where actually Wally Curran, who's the director of the Winship Cancer Center, was the associate director of the Cancer Center. But also learned, how do you run your lab and things that you don't learn when you're a grad student or a postdoc. In 2003, moved north and expanded my research program into NAD metabolism, which I'll tell you about in a couple minutes, and it sort of rose through the ranks of Cancer Center leadership there, where I was associate director for basic sciences at the Norris Cotton Cancer Center. And the research program also really flourished at that time. 2009, I was recruited to the University of Iowa. This is our new performing arts building, and developed something there called quantitative targeted NAD metabolomics. And also did initiated a program on reversible DNA modifications. We published papers, but more importantly, the larger initiatives at Dartmouth, herding cats, 70 cats in the basic sciences of the Cancer Center, very similar in some ways to the IBB directorship. Everybody has an academic appointment in a department, and we were running scientific programs in which we're trying to have added value with shared resources and collaborations and a cancer focus. At the University of Iowa, we were able to hire 8 new faculty colleagues, all of whom are funded and have been promoted to tenure or will get tenure. I built the university's HTS facility, and then in response to a call for new interdisciplinary clusters that would address grand global challenges of the 21st century, I wrote the proposal for the Obesity Initiative. And we're very pleased that not only did we hire 16 faculty members in 5 different colleges, many of whom have the only NIH grant in that college, for example, business and nursing, but we also united the research and outreach activities of about 60 existing University of Iowa faculty members that never had a nexus in which to talk about the problems of overnutrition, obesity, diabetes, and associated conditions, and all of their implications for human health, policy, law, et cetera. That wasn't my first rodeo in terms of tech transfer. At all 3 institutions, we've had invention discoveries that have been licensed. This is a little molecule that was co-developed with molecular probes that has continued to be sold by Fisher, that's useful for basically analyzing RAS protein enzymology. This is our basket of intellectual property at Dartmouth around nicotinamide riboside. This one is particularly valuable and generates a lot of income for Dartmouth. And then at Iowa, I have some new technologies that are either have been or I think shortly will be licensed. So now I'm going to spend just a few minutes telling you what I do in the laboratory in my abundant time. So this molecule on the left is called nicotinamide. This is nicotinamide adenine dinucleotide, NAD. From now on, I'll probably say NAD. NAD is the central coenzyme in metabolism. What do I mean by that? Well, consider that whenever we're burning fuel, protein, fat, and carbohydrate, NAD is what's called the hydride accepting coenzyme that allows our fuel to be burned, that allows our fuel to be oxidized and turned into CO2 and biological energy. So NAD picks up a hydride in these reactions, becomes the reduced form, NADH. This is reversible. This is called redox biology. So NAD plus is interconverted to NADH. NADH is reoxidized to NAD plus, largely at the inner mitochondrial membrane to generate ATP. So biological energy involves converting our fuels into this stuff called ATP, and it's all about these interconversions between NAD plus and NADH. NADH is also reoxidized overnight when we maintain our blood glucose and when we produce ketone bodies. Now, I've just changed this NAD structure a tiny bit by phosphorylating the 2 position, generating NADP. Don't worry, I won't take you too far down the road in biochemistry in these few minutes. But NADP is in a redox pair with NADPH, and that is required for us to make RNA and DNA. And NADPH is reoxidized in free radical detoxification as well as making lipids and steroid hormones. This is really important stuff. This is vital for every cell, every tissue in the body. And it turns out that not only are those reactions between NAD, NADH, NADP, NADPH just interconvertible reactions, but NAD is broken down. NAD is broken down in what we call signaling reactions. In the case of sun damage and DNA damage, NAD is used to generate a polymer of something called ADP ribose. And so now you've degraded this critical coenzyme that you need to oxidize fuel and generate ATP. That gets converted into a signal for DNA repair. There's another signal for calcium mobilization called cyclic ADP ribose that is formed essentially by degrading NAD. And then there's a class of enzymes called sirtuins that mediate kind of nutritional signaling. This is like belly full, this is belly empty, that do some biochemical reactions also at the expense of NAD. So NAD can be broken down, and NAD is required for our fundamental metabolism and vitality. 100 years ago in the American South, there were 1 million people with this condition, pellagra. They presented with diarrhea, dermatitis, dementia, and ultimately death. This is a deficiency of NAD precursors. And if you look at the sun-exposed skin, which is where this dermatitis is fulminant, that's where the sun is damaging her DNA and her NAD is being committed to these repair processes. And tissues are really unhealthy. Her gut is unhealthy. Her skin is unhealthy. Her brain ultimately becomes unhealthy. And it was discovered that fresh food can basically prevent pellagra. And in 1938, someone named Conrad Elvehjem discovered the 2 compounds that we call vitamin B3, nicotinic acid and nicotinamide, as pellagra preventative compounds. They're all precursors of this wonderful coenzyme NAD. So I was working, in 2003 in my laboratory, and I was trying to understand the structure and function of this enzyme QNS1. And if you look at this wiring diagram, tryptophan is over here. It has to go through 8 biosynthetic steps. There's 6 of steps here, 7th and 8th biosynthetic steps to make NAD. And then this is nicotinamide. This is one of the vitamin B3s. And this is the other vitamin B3. And people thought that they knew everything about the way NAD is made. And I basically didn't trust this received wisdom. In the course of doing experiments to figure out how this enzyme works, we discovered how the pathway really works, which means that we discovered missing players in this pathway. So we discovered nicotinamide riboside as an unanticipated vitamin precursor of NAD, and Pavel Bioganovsky discovered, and I discovered nicotinamide riboside kinase 1 and 2, actually, one in yeast and 2 of them in mice and people, that convert NR into NAD through this nice shortcut pathway. It turns out there was more stuff to be discovered. Wonderful graduate students discovered transporters, additional biosynthetic steps, another metabolite, retrograde pathways. NMN is very important for those of you who are interested in mitochondrial NAD, which is a super important topic. And then it turns out that there's a bunch of disease relevance and human health relevance in this pathway. So this enzyme is the major recycling pathway. This declines in aging, whereas our pathway is actually induced by a number of kinds of damage, and I'll show you briefly some evidence for that. And then this pathway is mediated by our microbiome, and this pathway is encoded by a gene that is neuroprotective, and this enzyme, SARM1, is actually a neurodegenerative factor. So it's pretty clear that there's potentials to modulate aging, response to nutritional status, and metabolic stress, and neuroprotection, and neurodamage by understanding more in this pathway. That's what kind of makes me tick as a researcher. So, so this is what our first data look like. This is the molecule. This is our vitamin. And unlike the discovery of the 2 previous vitamin precursors of NAD in 1938, where you had to basically give dogs a form of malnutrition, we figured the whole thing out in yeast genetics. And we figured out which genes were required, and then those genes are conserved in human beings. And so this was kind of our first fundamental discovery. In 2007, we showed that we could extend lifespan in yeast cells with nicotinamide riboside. Yeast cells live a very short life in high glucose medium. If you calorie restrict them, you can extend their lifespan. If you give them nicotinamide riboside, you can also extend their lifespan. This was a kind of amazing structural discovery that we had. I solved the crystal structure of nicotinamide riboside kinase 1 with nicotinamide riboside. For those of you chemists in the audience, you can see the amino group here and the carbonyl group here of the nicotinamide moiety. And it really surprised me that there was no specific recognition of the oxygen, that you could spin this around 180 degrees. And we said, gee, I wonder if nicotinic acid riboside is a thing, if it's a metabolite that could be used by nicotinamide riboside kinase. It is. And so we actually discovered a metabolite by doing structural biology, not an everyday occurrence. That was a red letter day. So, so why is this vitamin valuable or any different than the other ways that tissues can make NAD? One is that these NRK genes are active in all tissues that we've looked at. High-dose niacin is used to control cholesterol, but it causes flushing, and this is not a problem that we see with NR. High-dose nicotinamide inhibits one of the enzymes that is important in metabolic regulation and lifespan. NR does not have the property of inhibiting those sirtuins. It also costs the least ATP to make NAD from this pathway, and whereas some of the other pathways are turned off in disease, obesity, and aging, the NRK pathway is turned up by stress and a signaling molecule called AMP kinase. So, so there are mechanisms that challenge NAD, which I call nearly inevitable forms of stress. So alcohol intoxication, when our liver metabolizes alcohol, it converts NAD plus to NADH, and then the next fuel that we eat, the next food that we eat, doesn't get turned into ATP, it gets turned into fatty liver disease. Overnutrition, our lab has used quantitative metabolomics to show that if you overfeed a mouse, you depress its NAD pH, it can't control reactive oxygen species. noise-induced hearing loss. There's a decline in NAD, sun and oxygen damage for reasons I've already told you. Changing time zones, irregular hours. So there's a circadian cycle of NAD, neurodegeneration, heart disease, and it's widely reported that in aging, NAD declines as well. So I often get asked by somebody who's working in their lab, and this shows how I personally interface with other research groups worldwide, is, gee, I wonder if NAD is dysregulated in my favorite disease or condition. So when I was recently at Stanford, there was a professor in the audience that was interested in immune activation, and there was another group that was interested in myalgic encephalomyelitis, and basically they always want to know, what's the interface between NAD and my, favorite disease. And basically we have two paired technologies that answer that question. One is the NAD metabolome, which we can measure quantitatively by LC-MS, is it dysregulated by tissues from that disease? Or in a condition of immune activation, you isolate the cells, do they have weird levels of NAD metabolites? And then the gene set, we finished the gene set, so by targeted transcriptional analysis, you can see whether there's something going on with NAD. This particular figure just shows that in calorie restriction and aging, there's an effect on NAD metabolism. So we interface with a lot of groups around using these technologies. Just briefly, until I show you translation and then ideas of what could be done here. This is a model of a mouse model of obesity and type 2 diabetes, and this shows neurodegeneration in obesity and type 2 diabetes. This is the density, the intraepidermal nerve fiber density in the hind paw. So this is basically diabetic nerve loss that you're seeing here. But if you give these same fat or diabetic mice nicotinamide riboside, you can protect the nerve density. These data show that quantitatively, and you can also affect the nerve function. So they don't become insensate to temperature. This is, we have some mechanistic ideas into this, and we've completed a first clinical trial in obese men. This is a female rat who's been given paclitaxel, so a breast cancer chemotherapy drug, to induce chemotherapeutic neuropathy very rapidly, much more rapidly than overfeeding a mouse. So in a couple of weeks, they become hypersensitive to this touch test. But if you give them nicotinamide riboside, you can prevent that. You can also reverse chemotherapeutic neuropathy. And we've been funded by NCI for a phase 2 clinical trial on women with breast cancer in order to test whether this works in people. And then I won't belabor this point, but there's a group in France that knocked out their favorite gene in the heart. They are getting dilated cardiomyopathy and heart failure in this mouse model. And they call me up and they tell me nicotinamide riboside kinase 2 is the most highly induced gene in the failing heart. So the heart is failing because they knocked out some transcription factor. Nicotinamide riboside kinase 2 is going up. I said, gee, I bet NAD is going down. NAD is going down. And it turns out that not only in mice, but in human heart failure, nicotinamide riboside kinase is going up. This other NAD gene is going down. That means that the tissue is looking for NR in order to get its NAD back. So nicotinamide can't get the NAD back up. NR can. The reason is that the nicotinamide riboside kinase 2 promoter is responding to the bioenergetic crisis in the failing heart. ATP is down. Something called AMP is up. AMP kinase is activated. The NRK2 promoter gets turned on. And if you give this mouse nicotinamide riboside, you can see if you're a molecular cardiologist, the injection fraction is down in the disease, fractional shortening is down in the disease, and all of these parameters are improved by NR. So it has a lot of potential in diseases of metabolic stress. This is something I won't go into, just excitotoxic brain injury, so central brain injury also. In central brain injury, NAD goes down. Nicotinamide riboside kinase genes 1 and 2 are transcriptionally induced, NR protects. So translation. So in one slide, our experience with translation was kind of like dating, where you might have to kiss a few frogs. So initially we had the wrong commercial partners that were not going to develop it or didn't have the skill set to develop it. We then found our prince. They set out to do a clean, scalable synthesis and get a patented crystal form. We did animal toxicology. We did a human clinical trial to show safe oral availability and published those data. We got 2 FDA notifications for generally regarded as safe and new dietary ingredient. We're a consumer product now, and we're also doing more clinical trials for disease and conditions, as well as we're resisting attacks by pirates, which is unfortunate. Everything about this was surprising to me. You don't know how to do it until you do it. Now here's a kind of interesting problem, which is that this is available over the counter, right? It's a vitamin. It's a natural product. We found it in milk, and we showed it's generally regarded as safe. And it says on the bottle, not intended to treat a disease or condition. But in all of those animal models, I'm showing you what it does for the sick animals. What it does for the really healthy animals on regular chow is modest, right? And so the preclinical data suggests more benefit in diseases and conditions, but drug companies don't want to spend money to test it because we made this compound too inexpensive for consumers. Isn't that amazing? It's expensive as a vitamin, it's $500 a year, but drug companies want something that's tens or hundreds of thousands per patient per year in order to develop it. So it's really kind of a conundrum for me because I really want this to achieve its full biomedical potential, which is going to be in conditions of metabolic stress. So we have 2 solutions to this. And the first is a combination of something that's related to NR, not NR itself, and another compound that is strongly neuroprotective. And we think that there's going to be compositional IP that will allow this to develop that as a neuroprotective. Second is that we ventured into postpartum as a condition of metabolic stress that we don't consider a disease or condition. And it's actually really cool what we're doing there. I won't go into those data other than in a summary slide. That we found NR in the pathway, and this is an orally available vitamin through a constitutive inducible biosynthetic gene with some advantages over the other B3s. In animal models, there's resistance to weight gain, fatty liver, neurodegeneration, and heart failure. It's safe in humans. We're doing clinical testing for other diseases. And then just a teaser, if I come back and do a real scientific seminar, I'll show you that it boosts lactation and weight loss in a new mom, in a mouse and rat. The offspring have strong advantages, including physical performance and learning, and with like some neuroanatomical benefits. And actually the milk from an NR supplemented mom has high levels of brain-derived neurotrophic factor. So there's an amazing future, I think, of this project. But the other take-home is that our core technologies of quantitatively looking at the NAD metabolome and having the gene set basically tell us all of the diseases and conditions in which NAD is somehow dysregulated, and there's a therapeutic angle for us. Now, and thanking the people and the funding sources and collaborators. So now IBB. So when I look at what you're doing, I see a lot of really wonderful things. And I want to call out some of the things that you're doing, or I think you might be doing, or I would be, want to lead if I were to be selected to come here. And so first, a lot of budget on the core facilities because these things are expensive to run. You need very skilled people, such as many in the room, to operate them. And it wouldn't work for these people to have 10% effort for 10 different PIs. And that just wouldn't work. So having them in central locations with some subsidy, generous subsidy from the EVPR and management through IBB is really, really important. Your seed grant program, I'm sure, has a great return on investment, and it's fantastic that you get people together from different disciplines and you allow them to re-up when they write proposals, and that's terrific. Award nominations, this is something our department spends a lot of time on. I'm constantly nominating our faculty to be recognized, nationally and internationally, as well as, in the university. And then travel awards, you have a lot of poster sessions and grad students and postdocs. You can send your students out as ambassadors for your institute, which is a really great way to advertise it. The director makes a lot of introductions, kind of catalytic introductions of people that might not know about each other's programs. We have chalk talks in addition to seminars. So these are chalk talks that are largely designed to help people think about how to write a new proposal, how to develop their specific aims. Some of our chalk talks are faculty only, so that we can really get in the weeds and kind of criticize each other a little bit. Annual retreats, travel awards. I have up here grant pre-review. And this is something that we spend a lot of effort on in our department, both at the level of reviewing people's specific aims pages really early on, and then paying generally someone that has just cycled off a targeted study section to review one of my colleagues' grants before he or she sends it in to NIH, NSF, or another agency. So grant pre-review is a great activity to increase people's success rates. Training outreach, you're doing a wonderful job. And then I think sabbaticalship is really good. Visiting scholar program could be useful. Your regional and international partnerships, alumni and industry relations, super important. And finally, venture school, which I went to venture school, and I don't know if you have them here, but they're good to expose your faculty and your trainees to the ideas of building a business model and what is licensing and so forth. So some of the strengths of the IBB, and what Bloomberg said to Rothman. Paul Rothman was the dean that hired me at University of Iowa, Carver College of Medicine, who went to Johns Hopkins to be the dean and CEO of Hopkins Medicine. Chair of the search committee was Michael Bloomberg. Michael Bloomberg said to Paul Rothman when he arrived, "We got a good thing going here, Paul, don't screw it up." So that's kind of one thing I think a new director should keep in mind. So you've got great trainees, faculty, and I should say staff as well. I mean, a pretty diverse student body, and really commitment to diversity of the faculty and staff, as well as incredible quality. A great location here in Atlanta. You're not allergic to applications and commercialization and outreach. So some pure biochemists think that I become dirty when I discover a vitamin and then find that it's useful in human health. And sort of tech schools and places with engineering don't tend to have that allergy to application. So I think that that's really an advantage. Your preeminence in bioengineering is certainly something that the Institute should try to maintain and build on going forward. And I love the kind of vision statement of creating the next. So opportunities here. One is to increase visibility. I mean, I didn't know how great it was until I started looking and looking at the website and visiting here in person. Increasing the visibility to talent, to funding agencies, and I think to capital markets as well. Since you're not in Boston, used to be Boston, San Francisco, and now there's tons of biotech in New York, Seattle, and Southern California. And I think creating the next biotech hub in Atlanta is a absolutely achievable goal. But part of it, involves work to show what's here. Building in interfacial areas. if I were to come, probably my neighborhood could be called, if we were to grow something, we might call it something like metabolism and bioenergetics. I think that we would interface with cancer. I think we would interface with neuroscience. We would interface with bioinformatics, drug discovery, delivery, and almost everything else. We would need to think about metabolomics services. I would bring targeted NAD metabolomics. But in addition, other growth areas in bioengineering and biosciences, multi-scale modeling, certainly informatics. I'm not suggesting that you don't know this, right? You're putting up buildings that have informatics. But there's also some really interesting interfaces between sort of mobile, assistive, diagnostic, biometric technologies that where, you know, our mobile phones, you know, are collecting data that's going to be integrated with our genome, potentially our metabolome, our sleep, and you know, there's going to be ethical and social and policy issues of that we could involve other people at Georgia Tech in as those things are developed. Because as you do translational biology, translational medicine, you run into societal issues and privacy issues that have to be thought about early on. I think that there's an opportunity to expand partnerships in biomedicine with Emory. You have a really unique Department of Biomedical Engineering, of course, with Emory. I'm on a first name basis with the Cancer Center director there and the head of pathology as well. But I think that also at Georgia Tech, you could potentially do more with computing and business as you grow biosciences. So finally, I have an idea that has to do with visualization of our scientific discoveries. So, you know, VR and AR, virtual reality, augmented reality, and some of these technologies are essentially available with Google Cardboard around a mobile phone that we all carry. Some of these things can really disrupt scientific communication. So you can imagine, you know, Raquel Lieberman's student solves a crystal structure and goes and talks to Janet Murray's student about how do you create an immersive experience in which to learn what's going on in this crystal structure. When I mentioned this to the staff that set up the posters in the IBB atrium, that really liked the idea that there could be Google Cardboard instead of all of those things that we have to schlep around in poster sessions. But I think that ultimately the interaction between technology, the people, the undergraduates that are going to go into gaming, some of them can think about the 4 dimensions that occur in biology. We have X, Y, Z, and time, and we have a lot of things that interact dynamically. And so I would love to get involved with digital humanities and computing in ways that will make us really a global leader in education, research, and science communications. So that's what I got. And I want to thank you for your attention, and I'm super happy to answer any of your questions. Okay, so now we'll open up the floor to questions. And I think there are microphones that can move around. Yes?
I had a question about the venture school. Could you just talk more about how that is run? Is that just bringing in VCs to kind of talk about their experience and speakers and things like that, or is there a bit more to it?
Yeah, I learned in my last meeting that you're a site for NSF I-Corps, which is the venture school sponsor that I went to at University of Iowa. And ours was run it was coordinated by essentially somebody in the VPR's office, sort of your EVPR's office, and it had guest speakers from the business school. And we had, essentially, if you had a technology and you were thinking of creating a startup, or you wanted to learn about startups, you signed up for the venture school. And I think it was basically a free program that was open to faculty, staff, and students. And some of the people in the venture school were really quite advanced, like they really already have a startup, and they're refining their business plan. And other people were still at the what-if stage. And there were people like me that have a lot of experience in technology, invention that's licensed by somebody else, and relatively little experience in forming a startup. And my startup idea is not still fully formed. I think it's probably going to be in the NAD diagnostic space, but it actually requires more research in the lab before we can really spin it out. But it was really a combined activity of from the EVPR's office with B-School faculty, visiting speakers from the community who are mostly business school grads. And a lot of the people that were business school grads really like coming back to campus because they get an early look at technology that has not been poached on by somebody else already. Yeah.
How do we normally get NR in our bodies, and why don't we get enough of it, and what happens if you get too much?
We don't normally get vitamin B3 as vitamin B3. So we typically eat food, whole foods with a small w and a small f. You're eating cellular stuff, and cellular stuff has a lot of NAD in it. So, and then lesser amounts of NADPH, NADP, and NADH, right? And those compounds, all of which have 2 or 3 phosphates on them, break down into salvageable precursor vitamins, NR, nicotinamide, and nicotinic acid, depending upon how many enzymes and bacteria hit them before they are available to tissues. As we age and as we experience metabolic stresses, some of the NAD-consuming activities are in excess of vitamin salvage activities. And so in those conditions of metabolic stress in rodents, we can show definitively that chow doesn't have enough NAD precursors, and that nicotinamide riboside has better activity than the other NAD precursors in ameliorating the metabolic stress, largely because NRK2 is induced. In terms of humans, we've done safety data up to 2 grams per day, and the recommended, over-the-counter dose is 250 milligrams. And we're actually not the Wild West of supplements. So by having the FDA notifications, there's actually a registry at FDA that would collect adverse events. And there's, I don't know how many tens of millions of doses taken in 5 countries, and there's virtually nothing that's shown up. So we think it's really safe because it's basically degraded NAD, which we're exposed to lots of NAD. 2.
You want to go first?
Oh, there's...
Go ahead, Susan. How about that one? Okay, all right, I'll go.
And then there's the one over here.
So you briefly mentioned it, but do you have any more specific strategies about how to bring investors to the Atlanta community? You mentioned we're not quite Boston, we're not quite New York, and you gleaned over it a little bit, but I wonder if you had any specific plans in mind. And then the second question, what are your thoughts in terms of fostering and encouraging entrepreneurial activities amongst students and faculty in IPB?
Yeah. So I think that, having conferences where you invite people in the VC community and investment community here at campus is a good idea. You're absolutely a destination in engineering and bioengineering, right? And I think that I think the visibility is not fully there in the biosciences and bioengineering interface. And one of the last times I visited Atlanta was at Pharmaset. I think Pharmaset got bought by Gilead, and I'm not sure if they still have research activities here. But I think that over time there will probably be more employers that come here because the cost per square foot of lab space is lower than in South San Francisco and in Cambridge. But I think that until that time, I would want to try to hold conferences to get people to come down here, use the alumni networks. I do a bunch of consulting in Boston and the West Coast, and I think that it will happen. I've heard over the course of the couple days here that some of the faculty startups have launched in Boston, and that there may be some that are going to be launching closer by. So if you're building an incubator space or a research park, that's going to definitely help. It sounds like in computational science, there's a new building going up where the university and companies are going to be joint. So I think it's going to develop. Second question: how do you involve students?
Just foster those activities amongst faculty, students, so forth. We want to get into that, but don't quite have the know-how yet.
Yeah. Well, it would be great if you could, pair, business school students with students in the biological and biomedical and bioengineering sciences to just one way. I think I glossed over it in the talk that had the leaves on the tree, but coffee is a really important thing, I think, and getting people together from different organizations and maybe speed dating. I would try a lot of things. You know what I mean by speed dating, where you have kind of like a line of people on different side of the table, where you would basically, here's what we're doing in our lab, and here's what I think it might be good for, and then some sort of fintech or organizational management person on the other side of the table is cogitating about that, and they may or may not come up with something to do together that day, but they've created a connection, and that could lead to an interaction further down the road. I think that proximity and interdisciplinarity is really key, and the nice thing about the IBB, in which you try to get people unsiloed, is really a good structure for that going forward.
Okay, I have two questions.
Uh-huh.
One is related to your research. Have you ever looked, when you did your mouse studies or mice studies, how this affects the microbiome and the bacterial population in those mice, because they have an NAD metabolism too?
Yeah.
Did that get affected, or did you see a different population that is then promoting the health?
Yeah, it is different, and we've not published on it yet, but we're interested in it. There's effects on the gut that seem to be mediated by the microbiome, and there's some other effects on the gut that seem to be host. But yeah, it's a good question.
And the other question is, when you, from your experience doing the high throughput facility
Yeah.
in Iowa, what do you foresee that can be done to expand more facilities here at IBB?
Part of it is explaining how important it is to everyone's competitiveness, right? Because there's two types of there's two administrative philosophies with respect to cores and those kinds of things. One is that it's nurturing our faculty, it's investing in our faculty, and the other is, it's hemorrhaging our money, right? And you would never say about your grocery bills, you're hemorrhaging, right? You're feeding your kids. And so, so it's very important to be in those conversations with leadership so that they understand we can't do what we need to do without those cores. I think it's important for them to be not duplicative with other things that are available elsewhere. We might get to be really, really good in some things that Emory, for one, can't do, and we might let Emory do our samples in certain areas. And if you have those kinds of business arrangements and MOUs with other organizations, it's going to reduce the cost to our EVPR, but allow our researchers to have access to things that are cutting edge. It's also great to have, corporate customers that you can charge kind of top dollar to, but, you have to maintain a level, and affordability for the community. Yeah.
I have a science question.
Okay.
I was wondering, with your NR,
Yeah.
what does it do to lipid profiles in metabolic syndrome patients or animals?
We don't entirely know. The problem is, so is it going to be the next niacin that doesn't flush? That'd be huge, right? So,
Company I've worked with before.
Yeah. So we don't really know. So as you probably know, rodents are not a good model of human dyslipidemia. They carry most of their particles around as HDL, right? Even if they're overweight. Most of the initial trials you have to do with healthy human adults, right? Now we're starting to do things with people that are overweight and type 2 diabetic, and we hope to be getting those data. There's some preliminary, indications of, clearance of, hepatic steatosis, but HDL, LDL, and triglycerides, I don't know yet. Yeah.
So in your CV, you had mentioned an article that is in process.
Yeah.
In early stages, academic money.
Yeah. What does that mean?
I'd like for you to share with us what that means and what the principles are.
Right. So this should hopefully be resonant at Georgia Tech. So metrics, right? So it's easy to evaluate some things. It's harder to evaluate other kinds of professorial activities. So it's easy to measure student semester hours, right? And then if the only metric is how many student semester hours did you deliver, then everybody in the department wants to give, really large lecture classes, right? It's easy to measure grant dollars. It's easy to measure impact factor, what exactly it measures, I don't know. But it's not easy to evaluate all of the things that we do. And so, and you can't dismiss any of the missions in an academic department. You're doing research, teaching, and service, right? We're serving on each other's graduate student committees. That time is not really compensated time, but I need my students to be well advised. You need your students to be well advised, and so we advise each other's students. So I don't have wins over replacement player or so there are these new metrics in baseball where you try to figure out the value of people, and it was kind of popularized in the book Moneyball, where I think it was Billy Beane discovered that people are overpaying for batting average, and walks are as good as a hit if you end up scoring runs, right? And so I think the idea of academic Moneyball is that we find ways to measure people's impact, to respect people's contributions in a department, even if they have unique metrics. So like a relief pitcher in baseball is incredibly valuable, but only comes in certain situations. There's a left-handed batter in the dugout that has to come in certain situations. And the whole organization, may not be able to work without all of those different pieces. I'm not sure that I, can. I think that the goal is to provide department chairs and deans and more senior administrators ways of thinking about the faculty that allow people's contributions to be recognized no matter what it is that they do.
We have time for one more.
I have a technical question. I think we are thinking back and forth in technical and specific questions. So you mentioned that this looks like the future of your work lies in this quantitative analysis. For example, you've done LCMS.
Yeah. Yeah.
And so I do quite a bit of LCMS for a living. So whenever people mention quantitative LCMS, I drop them very wrong way. Anytime you talk to analytical chemists, and I'm not an analytical chemist, they would yell at me when I say, well, I've done actually quantitative LCMS, because that implies full illusion of an internal standard.
I have that.
And every single condition identical in the buffer because all the charge pathways in the electrospray and charge suppression, which part of the fluid sample all has to be there. So it's usually an analytical field with a fit. That in the LCMS, your best really like to be doing identification, discovery, and maybe what is semi-quantitative comparison, kind of up and down. So if you can comment a little bit on how sophisticated those workflows are.
My data are on an absolute scale. We do our standards in matrix. We have internal standards. We know what % loss we're taking. We don't report NADPH and NADH because we can't reliably quantify them. So it's not just like looking at a microarray where it's up down. I have competitors that will get all excited about twofold differences in nicotinamide, but the level of nicotinamide doesn't rise to a level where it would inhibit any enzyme. So we're very cognizant of those issues, and I think that our data are pretty solid. The other thing that just to get back to Susan's question that relates to what I call academic moneyball is that we have to be really sensitive to faculty activity, right? Because like administrative burden of faculty doesn't help anybody, right? Because what we really need to do is we need to protect our creative time so that we can do the high impact research and so that we can do really quality education. So it's really important that our incentives are aligned so that the students are getting the best education. We're in compliance, with the research office. We're spending, dollars responsibly, but we also have time to think and to do our unique work, because if we don't, do our own work, then it just won't get done.